All right, so in this video, we'll be talking about the cytoplasm, still in Unit 1 in anatomy, going through the cell. And this topic will be a lot of stuff that you've heard before. There'll be some new stuff maybe, but for the most part, it's just going to be a review of what you already know. And so still useful to make sure that you kind of remember things as you go through this. So what is the cytoplasm? The cytoplasm is the material between the nuclear membrane and the plasma membrane. It is all the stuff that is called cytoplasm collectively. This includes three things. This includes the cytosol, which is the actual fluid portion, the organelles, which we're going to spend some time talking about those organelles, and the inclusions. Inclusions may be a new thing. And inclusions are just chemical substances that are present in the cytosol. These will differ depending on the type of cell that it is. So the cells are very specialized, of course, to do a specific task. And so these inclusions are going to help with that task. So three things in cytoplasm, and there they are. Now we're going to talk about organelles. Before we go on, most organelles have a membrane in them. So very similar to the plasma membrane. And this membrane allows them, just like the membranes in the cell, just like the skin and the different structures in our body, the membrane allows that organelle to have a different internal environment than the rest of the cell. So it allows it to complete its specialized function as well. And so the first organelle that we'll talk about is everyone's absolute favorite, voted number one the last 7 years in a row, actually took over ribosomes 7 years ago and really hasn't let go of that number one spot. And this is the mitochondria. Mitochondria break down sugars and other molecules to convert those things into ATP. ATP is the energy currency of the cell. The cell uses ATP to do lots of various processes. A couple things about mitochondria: they have a double membrane. As you can see here, you have this dark orange membrane and then the light orange membrane that's all folded up on the inside, which gives that organelle lots of surface area. And mitochondria also have their own DNA, and they have their own ribosomes as well. And so they act and reproduce independently of the rest of the cell. Now, they still require the cell to do things. So much of the mitochondria's processes require proteins, for instance, from the main nucleus. And so this isn't like they're their own independent organisms, but they reproduce on their own, and they have a more of an independence, and they are the where all the energy is produced inside of a cell. Next is the ribosome. The ribosome is the site of protein synthesis. This is where proteins are made. Proteins are important because they carry out most of the cellular functions. They are not bound by a membrane. And so sometimes you'll see ribosomes referred to as cellular particles because they're not like this nice little membrane thing. They're just this little bitty thing, and they're really small compared to the rest of the organelles. You can see the ribosomes here for the mitochondria. See how small they are compared to them. And a cell may have several dozens and dozens of mitochondria. So these are very small little particles. They're made up of two subunits. Don't really need to know any of the ins and outs for those subunits. And they can be found freely in the cytoplasm, being just kind of roaming around, or they can be found bound to our next organelle, which is called the endoplasmic reticulum. So you can see here on the endoplasmic reticulum are these little red dots. They're not there for decoration. They are called ribosomes, and they're actually doing a thing. They're making proteins, still the same function, but they're just making them on the ER. So that brings us to the next point. There are two kinds of ER. There's rough ER, and that rough ER has those ribosomes on them, and the smooth ER, which doesn't really have the ribosomes on them. So they have two different functions based on that. The rough ER obviously is where the ribosomes are, and the rough ER is where the proteins are folded. Proteins are three-dimensional structures. We're not going to get into how they're folded and all that. That's for a whole other class. But these proteins basically fold up inside the ER, and they wait to be packaged and sent to the Golgi apparatus, which we're going to talk about in a second. and they're packaged in these little temporary storage vessels called vesicles. I'll show you those too in a second. Then next is the smooth ER. The smooth ER is the site of, it does a couple things actually. The site of lipid synthesis. So this is where like phospholipids for the cell membrane are made. This is where a lot of cellular detoxification occurs. And so organs that deal with more detoxification are going to have more smooth ER, kind of like a liver or something like that. And then in certain cells, they have a specialized function where they store types of ions. And so we're going to talk about in muscles, they have a specialized smooth ER that stores calcium ions, for instance, and we'll get there in a later unit. But so the smooth ER can have different types of functions, whereas the rough ER typically just does that one thing. And that brings us to the Golgi apparatus. Not part of the ER, it's its own thing. Sometimes you'll see it called the Golgi body, but I see that less and less now. So the Golgi apparatus receives proteins in vesicles. You can see that kind of here, transport vesicle from the rough ER. So imagine it's like a little package that's going to the Golgi. It's going to bind with the Golgi. It's got those proteins in there. And the purpose of that is to receive those proteins and then modify them. It modifies them with some sort of carbohydrate or something that like tags it, because those proteins are meant for secretion out of the cell. And secretion just means that when a cell releases some sort of material. That's where you see this secretory vesicle here. These proteins have gone in, they've been modified, and they are ready to go out and do their job. All right. This whole thing, this whole rough ER, Golgi body, or Golgi apparatus, and vesicle, all of that is typically referred to as the endomembrane system. We're not going to get into a whole lot of depth with that, but I think it is important to understand that these things are working together to produce proteins that are going to be leaving the cell, and that is their main function. Next are organelles called peroxisomes, which may be new to you, and then lysosomes. Peroxisomes sounds like peroxide, right? So think about it that way, because that's actually what it does. So peroxisomes are little organelles, and what they do is they go around and they neutralize substances called free radicals in the cell. A free radical is an atom or a molecule that has some spots open in its valence shell. If you haven't had chemistry, the easy way to understand this is it can bind to things pretty easily. And so if you can imagine this, having this rogue molecule floating around, binding to things, and it may not need to bind to those things. And so it can tear stuff up, which is just a really crass way of thinking about it. So think about these little things floating around, and they may be trying to tear things up. And so the peroxisome is going to go in there and it's going to neutralize those free radicals. Now, when it does that, it turns them into hydrogen peroxide, which is also not a good thing for the body to have in it a whole lot. And so then the peroxisome will then take that hydrogen peroxide and turn it into water and oxygen, which are perfectly good things to have inside of us. You've probably seen this reaction anytime you've poured peroxide on a wound. The bubbles are not your wound getting better. It is the catalases inside the peroxisomes that are causing that reaction. The bubbles are just oxygen being released, and that what's left is water. And then there's lysosomes. Lysosomes basically their goal is to digest foreign particles that are inside the cell. And so some cells that their whole job is to go around and eat stuff, which like immune system cells, they have a lot of lysosomes in them. Lysosomes can also be used to digest old cell parts, excess materials, basically anything that needs to be digested. The lysosomes are going to be doing that. They're almost like little stomachs. And so the inside of the lysosome has a much higher pH or much lower pH than the rest of the cell. And again, that has to do with that membrane bound internal environment that we were talking about. Next is the cytoskeleton. Not really organelles, but its own kind of thing. Cyto means cell, skeleton means skeleton, and so you can kind of get what's going on here. There's 3 different cytoskeletal elements that we're going to look at, and there they are before you. There's the microfilaments. These are the smallest, hence the word micro. And these are made up of a protein called actin, which we will talk a lot about in the muscular unit. These are responsible for cellular movement. And so when a cell is actually moving around, like muscle cells do, for instance, the actin filaments are what are causing this to occur. And these aren't permanent. They're constantly being made and unmade. There are permanent parts of the cytoskeleton, which we'll talk about in a second. But it's important to understand that this is a fluid kind of thing. The intermediate filaments are not so fluid. All right, they're like it says, they're tough and they're insoluble. They're not able to be broken down. And so think of these as being more permanent portions of the cytoskeleton. They provide a lot of structure for the cell. And then microtubules are more like ropes. They're like hollow tubes. Think of like a straw or something, but think of a flexible straw, like that candy that you can get that's really just not good, but people like it. That's like a tube that has a bunch of crap on the outside of it. But microtubules are like those tubes without all the crap on it, the salt, sour mess, whatever. Getting off topic. So microtubules, think about them as being little ropes, and their purpose is to determine the cell shape. And so they can kind of pull the cell into a certain shape. So they're very important during cell division, which we're going to talk about in another video. And they're also important for moving things around inside the cell. So like if I was to attach a rope to one of the desks in my room and pull, I would be able to change the location of that desk. And so microtubules have that same kind of function. There are some specialized microtubules. One of them is called a centriole, and the centriole forms during cell division, and their purpose is to move things around. Again, they form as anchor points for the microtubules, so the microtubules can move things around. Think about all the microtubules that are attaching to chromosomes and doing all that kind of thing during cell division. They're going to be anchored or originated at the centriole. That's good enough for our purposes. And then there's some other microtubules that are called cilia and flagella. Cilia are typically these tiny little hairs on the outside of a cell, and their purpose is to move things across the cell surface. Think about like crowd surfing, right? When the dude jumps off the stage and the people hold their hands up and they're moving the dude across the crowd. That is what the cilia are doing. They're like the people's hands. All right. Whereas flagella is almost like a little whip that is whipping around and allows the entire cell to move around inside of its environment. And so those two things are typically the cilia and flagella. They're found on the outside of the cell, but they're still formed from these microtubule structures.